Monica. I'll be leading the webinar today and the meeting today. We have just 2 announcements ahead of our scheduled webinar with Mike Cherry. And so Dory Main will start, and then we'll have Rex Nelson. So Dory, can you go ahead and give your announcement?
Can do, Monica. Good morning, everyone. And so this is just a quick announcement about the progress of the AgBioData proposal for the Coordinated Innovation Network grant, which is due to be submitted. It was due at the agency October 31st. So we have pretty much a draft written. We're not quite ready to send it out to you right now, but it should come out to you tomorrow. At least all the approach and what we're asking the community to do will come out to all the list of the people that are on the email list to ask you to go in and add any comments to that document, not to make any edits. I think we're just going to make it so that it's comments at this point. Is there anything that we're missing in our approach that you think we don't have in there or you don't like? We'll also be at the same time also sending you a link to a Google Sheets document to ask you to go in, and your names are already all in there, just to ask you to select what your primary work group would be as part of this grant, and also to indicate your willingness to attend a yearly workshop, which would be funded through the grant if we get this grant funded. So if you could do that, that would be great. We'll use that information in our actual proposal. And the other thing is we would require letters of collaboration from those of you who are willing to come to the workshop, and we'll just provide you with a template, and it's just simply to say that you're willing to attend the workshop and participate in the AgBioData. In terms of the grant itself, most of the money is going to, it's a $1 million grant over 5 years. The majority of the funds will go for 3 components really. One, to fund a half-time coordinator position, which actually would be Jackie Campbell, who's a co-PI in the grant and part of the steering committee. The second one would go to do a sustainability study, which will be done by Phoenix Bioinformatics with Eva Mendez leading that. And the third part is for yearly workshops, so that we can meet in person like we did in Salt Lake City, and help us have these really targeted workshops. I think that's about it. That's all I really wanted to say about it, but I think we're on track with it, and you should see something tomorrow. Thank you.
Great. Thank you, Dory. Okay, and Rex, you had an announcement as well? Rex, are you there? You might be muted.
Yes, I'm muted.
Okay,
Lisa asked me to say a few words about my PAG workshop. And I also might add that Dory also has a PAG workshop. But my PAG workshop is entitled Big Data, Manage Your Data Before Your Data Kills You. And the reason that we decided to try to start this workshop, which we started last year, was to start or to allow a forum where people could start bringing issues of handling and reusing data to the forefront, and maybe people would start thinking about data reuse in a serious way. So currently we've got 6 speakers for this year. Sarah Odell, which is a graduate, or I think it's a graduate student. Lisa's going to talk. Lindsay Reuter, which is, she just got her PhD, and she's going to talk about normalizing expression arrays. Philip Bayer, which is from University of Western Australia, and he is from Dave Edwards' group, and he's going to also talk about issues that they've had when in performing multi-center trials. Ben Campbell from the University of Minnesota, he's a postdoc, and he'll be talking about issues that he's seen. We also have finally Peggy Ozias-Akins from University of Georgia, and she will also talk about issues that she's seen in statistical analysis. The subject matter is wide-ranging, and as such, we would accept anything, particularly from our community here, the biocurators. If there's an issue out there that needs to be brought to the forefront and have a conscious raising, please contact me and we'll put you on for next year. If you have a student that could come and talk, that'd be fine with me. So that ends my very shameless plug.
An excellent plug, Rex. I'll just quickly mention there's 2 workshops that we'll be having. One, of course, is the triple workshop that will be continued again this year. I'm not sure who's actually on that one. I'm not organising it. And the other one is an NRSP 10 workshop, which is actually a very project-specific workshop on these 5 databases that our group hosts. And we will post all this information from Rex and from Triple and any other appropriate workshops on the AgBioData website under conferences section that we either have or will add very shortly. Thank you.
This is Pankaj. Can I talk about PAC 2 workshops quickly?
Sure.
So I'm hosting 2 workshops there. One is a lead one is on systems biology and ontologies. So we are looking for a couple of speakers from how they have used the data for doing systems-based analysis and doing something public goods in terms of they have found some good genes and good candidate genes for phenotypes and QTLs, or even for issues on big data. So there's a lot of overlap, but I think focus would be on biology rather than on the data in these workshops. And then the other one, which I'm co-hosting with Kusum Nithani from University of Arkansas, is on genome to phenome, next generation sensors for sensing plants and environment. And we have quite a number of people generating new kinds of sensors and tools, and also the data that is associated with that and standardization. So, and not only these, but also training new generation of scientists for building innovative solutions. And so that's another workshop which if the group AgBioData would be interested in, let me know on that side. Thanks.
This is Sushma. I just remind that we will also have Grameen workshop as well.
Great. Yeah. Well, thanks for announcing these. And if anybody has any workshops that they would like to announce to AgBioData, just email the steering committee and we can post that on the website. Thank you. I'd like to move us along to the main event of this hour, which is a webinar by Mike Cherry. So Mike is a professor of genetics at Stanford University, and he's a key player in a number of influential databases and other projects. First off, he's the lead of the Saccharomyces genome database, also known as SGD, which is the premier genome database on yeast. He also leads the Encyclopedia of DNA Elements Data Coordination Center, or ENCODE. And the goal of the ENCODE project is to discover new functional and regulatory elements in the human genome. And the Data Coordination Center that Mike leads is where the data and metadata from this project is stored and synthesized for reuse by the scientific community. He is also a founding member of the Gene Ontology, which we should all be familiar with here, and he's a member of the Gene Ontology Consortium. And he is one of the creators of Regulome DB, which is a web resource for integrated knowledge on regulatory elements. And then finally, last but not least, he is a member of the Alliance of Genome Resources, which will be the focus of this talk today. If you haven't heard about it, the Alliance is a consortium of the Gene Ontology and also 6 model organism databases. And the aim of the Alliance is to integrate the efforts of all of these databases and the Gene Ontology in terms of shared data, standardization of data models and web interfaces, and unified outreach to stakeholders. So as you notice, there are a lot of parallels between the goals of the Alliance and the goals of AgBioData, and this is the reason that we've invited Mike to speak to us today. And we have a lot that I think we can learn from Mike and the Alliance, and we really hope that Mike's talk today will foster lots of discussion today and beyond. So thank you. Before Mike starts, would you guys please remember to mute yourselves and also note that this webinar is being recorded. All right, thanks. So with that, Mike, thanks for coming, and you can take it from here.
Great. Thank you, Monica. It's really great that you invited me here. It's actually nice to see so many names on the participants list that I've known for quite a while, from way back. one little thing that wasn't mentioned. So the first database, I've over the years created lots of little databases, and some have gone away and some have continued. But my very first database I made was on Rabbitopsis physical mapping, Cosmid clones, and that was actually funded by the USDA. So that was the easiest money I ever got. They came by 1 week, wanted a Rabbitopsis database, and I think we had a check in 2 weeks. So anyway, with any other funding source I've had. So, and that was in 1991. So that's how long I've been around. Yeast databases existed for 25 years. We just celebrated this summer with that. So I want this to be more of a discussion. I'll give you a little bit of an introduction into the Alliance. And I must apologize. I was coming down with a cold last night. So anyway, I may have to go offline for a second or two if something comes up. Also wanted to congratulate you guys on the white paper that just came out in database. That was very interesting, and I thought it was really well done paper, and talked about many of the issues that I think the Alliance is dealing with at the same time. So let me switch to my slides. Or slide, just one slide. Let's see. Okay, is that full screen?
Yeah, we can see it now. Thank you.
So two sort of significant things at the very top there. There's the URL for the Alliance site, and then there's this paper that just came out in the last month, which actually describes a little bit about the mods, but then some of the challenges that the Alliance has to deal with. So the Alliance is made up of 7 groups. There's 5 mods. So the Alliance is really a mandate from NHGRI, and a way that they started pushing us, say, 4 years ago about sustainability, that it was really a business decision, not a scientific decision, which is always sort of tough to deal with when you're dealing with science. But this is the way funders work. They felt they were spending too much money on mods and wanted to figure out how to reduce that. And so they made a lot of noise, starting like 4 years ago. It got worse about 3 years ago with things where they basically sounded like they were going to walk away from the mods. And I think this was really all about the director of the NHGRI, the National Human Genome Research Institute, who funds the Big Five. It was all about him trying to get other institutes to pick up the tab for us. Of course, that didn't work. Everybody says, well, you're doing a great job, keep doing it. And so it was left to the NHGRI to figure out what else to do. They realized they couldn't jettison us. And so what they've done is decrease our funding. So it's in a stepwise manner over 4 years, where we're going to drop to 80% of the funding we had 4 years ago. But they'll sustain us at roughly that level, they say, for the at least the next 4 years is about as far as they get on that. So that's really reducing the funding of the mods. But to help with that, they're giving us a little bit of money back, not much, but really seed money really to facilitate as we're a collaboration. And a couple of the key things that they wanted us to deal with, and in reality is things that we've always wanted to deal with and just never really gotten around to do it, unfortunately, was I think the primary thing that everybody always heard is that our interfaces were so different, you couldn't actually find anything from one site to the next unless you really spent that sort of steep learning curve to figure out how to find genes and things between the various sites. So one key thing is they wanted people that work across multiple model organism systems to be able to use the databases effectively without sort of learning these special languages, as it were. But I think more importantly, NIH being NIH, they wanted to make it easier for those that aren't working on model systems, for example, human, to be able to find the information in the mods. They believe the information, rightfully so, is very interesting and very important out of the mods, but it's not being distributed in a good way, provided in a good way. So really interface was a big deal, But of course, to have a common interface, you need common data types, you need to agree on curation standards. There's many things like that that come into play. So I think it was really 2 years ago, they gave us this small little supplement to foster us having meetings and be able to employ maybe 1 FTE at the various sites. So I forget the percentage-wise, but it's a small percentage of the total funding that goes into the mods. Maybe, I think it's less than 10% total. So, and that was just distributed, proportioned out over what the funding was for the existing mods. And so, and in a second, I'll get into these points here, but I think you could all appreciate that perhaps our biggest problem was a social problem. Is that the mods, and I didn't say them, so the 5 from NHGRI are yeast, fly, worm, zebrafish, and mouse. And then rat is funded by a different institute, but they were allowed to get a little bit of this money and come in. And then the 7th group is actually the Gene Ontology Consortium, which at one level is a little bit of a mismatch, but at another works out well, and I'll try to touch on that in a second. But the social problem is really that all of these mods have grown up themselves. I mean, like I said, SGD has existed for 25 years. And we became, you know, kind of very instilled in a particular way of doing curation. We've had lots of meetings through the International Society of Biocuration and things like that, where we have gotten closer, but our tools have always been typically different. And so one of our goals was not to state them too high, was that right now maybe we have 6 different ways of doing something. Let's get it down to 2 or 3. That would be a success, right? And so while we may have 6 different interfaces for curation, let's try to get it down to 2 or 3. So obviously 1 would be better, but at least, you know, that allows somebody to sort of step off to the side and not really participate on everything. And that's really the hard part, is that while the mandate would be to make, you know, one Uber database of things, the practicality of that is small in any sort of short time. I mean, maybe over, you know, 5-10 years or something like that, you could imagine more of that happening. But it's certainly just sort of too many moving parts to do that. One question that always comes up is, will the mods go away, and will there just become one big alliance site? And the answer is the mod projects will not go away, because of the specialized curation and expertise that each mod has with dealing with a particular organism. Particularly with the curation, that domain expertise is essential. So the projects won't go away, but it may all get combined into sort of one software interface database thing. You talk to different people, you sort of get different answers. That won't happen real soon. I mean, that, you know, certainly within the lifetime of the next grant phase, which is like 4 to 5 years, that won't happen. But, you know, you never know going forward. At one level, it makes a lot of sense, but at another level, there's a lot of very special data types. And if they already have a tool to do that, should they change or not? You know, that all comes down to prioritization and financial commitments as it goes. So, so that's sort of the big summary of some of the rationale. I do see this as a positive thing. You know, I mean, we had our arm twisted to do it, but it is working. I think we're getting over lots of the social problems. I think, you know, with any sort of collaboration like this, people were unsure where they should step forward. You know, they didn't want to step on others' toes. Oh, well, you do this better than I do. I'll stand back. You know, no, please, you first. You know, sort of thing. And then there's the software development was also difficult because many developers within the 7 groups use different languages. And so, you know, standardizing on a particular software language was, in a sense, a big deal, because if somebody used, you know, C, they wanted to use C or Perl or Python or Java. We had all 3. Which database product do you keep? You know, how are you going to store it? So all of these things have sort of turned out to be essentially who's going to do the most work on that component of the site, and they use that language. So not optimal, but at least it's moving forward. So any other thing just to start? So, and certainly interrupt me as I go along. Like I say, I just have really this one slide, and I know there's many pieces to the Alliance, and I certainly want to hear your questions. I think that would be a great way to move forward on what I have to say. So in this article, what I've labeled here, the Alliance paper in Nature, it goes into pretty good depth on these 4 challenges. So distributed location of data. So I've talked about the website, but even more important is actually the APIs. You know, these days, the modern way of making an interface, a web interface, is to create an API and having JavaScript do the rendering in the browser. So there's not really the old way of having everything constructed on the server and displayed back. So you really need a robust API. But there's a flaw if you actually make your APIs just to serve the website. You need to actually have all of your data available in some, reasonably transparent way, and build into that sort of a powerful query mechanism of some form. I mean, that's the goal. That's hard. And none of us has really done that well. I mean, some of us have done that in various ways. I think maybe Worm and SUD might be a little bit of a leader there. We're not at anywhere perfect. And then some of the mods really haven't done APIs much at all yet. So there's still more the classic way of making websites. So websites driven by APIs, then that allows the data to be used by anybody via the API. And I'm a big fan of really teaching everybody, all graduate students and undergraduates for that matter that are doing biology, having them learn a language like Python or something, because I really see this is the way they should be interacting with the websites. It's fine to look at a small list of genes or the annotations for a particular gene, but more and more, even as you guys were talking just a minute ago, there's many different presentations on big data, and you really need to be getting at many pieces of data from many different genes and putting it together to annotate these bigger data sets that you have and that the rest of the research community has. And so learning a little bit about APIs is actually really critical, because the query you need, you can program, and you don't have to, try to troll through a website to get at that. So APIs are really critical, I think, moving forward. That's one of the big things that really everybody should be learning. Okay, so unique user interface, I sort of talked about that. That's also a little bit of a hard thing, is trying to build the site. Right now, it's really just the intersection of the various data that we have, and we're not really gotten to the point where we have the unique data from an organism yet. That's clearly something that we really need to deal with a lot. The data formats, I mean, nicely for the bigger, Go interactions, orthologies, those are the data formats are pretty easy. Phenotypes is one that everybody's sort of done it their own way. Even though there's ontologies exist, there wasn't always one ontology that was used. Some groups made their own control vocabularies and such. So it's a matter of getting everybody in sync on that. And the thing that was accepted, I think, after some time is the realization that a group had to do some work on their data, right? You've curated it one way and everybody else did it another. Well, you need to spend effort to make it like the others. You just can't, it's just unreasonable for the community really to say, oh, we're going to keep doing it our way, and so it won't match with the others, right? To integrate data, you need to be using the same formats, the same ontologies and such. Right? I mean, the whole purpose of curation, as you know, is that the researchers say things in different ways, but you need to be able to integrate, all papers about a particular gene and describe the function of that gene, or the phenotypes, or whatever. And to do that, the curator's got to be picking from ontologies and actually describing that in similar formats. So we're not really doing justice to our users, our customers, as it were, of the data, if we're allowing somebody to sort of play their own game. And that was hard for a lot of people, and there's still work that's sort of on the list. I mean, you can imagine some people have 10,000 papers that were done in a particular way. Recurating 10,000 papers is not fun, but at least they have the annotations they created first, and it's really maybe just verifying something about the paper. You don't have to read the whole thing. It's a big job, but it's not maybe as bad. The standards there, and so there's always, right, there's standards and there's plenty of standards. So whatever, there's a joke in there somewhere. But we're really trying to lock on truly community standards, and in many cases something exists, instead of trying to create a special alliance standard. And that's always hard because I do it in a different way, and so why don't I add an extra column to this file? You've probably all seen that in many ways. And so that's been a little bit difficult to control, just because people want to keep changing the standard. And then scalability and sustainability is always a big issue with something that you're creating new, because you start out with a small amount of data and a small number of users, and then that can quickly grow, and then all of a sudden you realize that the way you created it just doesn't scale. So we're involving many different people to be involved with this sort of the data ops side of it. How can we actually scale the computers and the algorithms used as the number of users increases? So you can imagine if all mod users were to use the Alliance side at least some of the time, because that's where we're really integrating data, that's a very large number of users. And so we have to deal with that. So I think I mentioned some of these things maybe before, but so common curation tools. So by common software to display things and visualize things, you need to have common curation tools as well. That will help enforce the common metrics and the SOPs that need to be there to actually have truly the same data types used. Unique IDs, we pretty much do this already, but we might have used a separate ID for some external database. Again, it was just history. That's an easy one to sort of fix up. The development of tools for discovery and visualization. So many people are very clever about how they control through their own database, but we really need to extend that to other databases. And so, but how do we prioritize that? Those typically take a lot of effort and happen, really require a year or so to be created. Funding, okay, the funding decreased. What can we do to sort of work together and get other funding pulled in? The mods are supported by longer grants, like 4 to 5 year grant terms, but trying to get funding for 2 years, say, to add a new data type, metabolomics or something other, something we could do. It's then just a matter of doing it. That's a PI level sort of thing. So right now, and I probably forgot one or two here, but the data types that we have integrated or just about to be on the website, or orthology, phenotype, variation, human phenotype, aka disease. I know they're not the same, but that's one way to say it. Gene ontology and interactions. And so these are pretty much all ways that many ways that you could actually go from one organism to another. You could follow through a phenotype. You could try to look at the variations within a particular gene. You could look at the associations between orthology and genotology and such. So that was my overview of things, and I sort of want to leave there plenty of time for questions. I'm certainly happy to go into more detail, whatever you guys want.
Mike, I have a question. This is Lisa Harper. You mentioned the social issues, and I think we are having that issue as well. Do you have issues with your user groups? I mean, the people that use the databases, do they not want this to happen, or are you having to retrain people to go through the alliance, or how does that work? So
yeah, the social aspect was really about the members of the alliance. The users-wise, I haven't really noticed anything. Exactly like you said, the biggest issue from the users is that there's uncertainty. They don't know if the mods are going away, because that's what the NIH director implied at one point. And so there was this big campaign where they got 11,000 people to sign a letter, and it was sent to the director of NIH, Francis Collins. So I think that's maybe the biggest worry. And so at yeast meetings, I'm always asked, are you going to continue existing, sort of thing. So far, the comments I've heard on the website is that people like it and that they're able to find information, you know, they know the yeast information, but to get the connection into Synovitis, they're able to do that more easily because of the Alliance site and the way we put things up. So I don't think users have been a problem yet.
Okay, good to know, good to know. How do you interact with your users? Do you, through feedback or something like that?
So we have pretty active help desks where a lot of people come in. We try to do a survey once a year, so those aren't really great. But at meetings, you know, we create little booths and people come up. And so we had a yeast meeting 2 months ago, and you know, I would guess more than half of the attendees of the meeting were in one way or the other talking to the SUD staff about what they liked or needed and things like that. And we also have workshops at those meetings. And we go to, so those are community-specific meetings. But we also try to go to other meetings and have workshops as well. So partly that's outreach to everybody, other research communities. But it also is a time where we can show our features to people and get an idea of what they need. One thing, just as an aside, one thing that I learned a long time ago is that people will come up to you and say, oh, what's new, right? And so I'll be talking about the things that we just released in the last 6 months. But in reality, there's a feature they haven't used or even known that existed because they've sort of just switched the focus of something in their lab. And so we need to remember to ask them what they do. Yeah.
Yes, that's right. We have that too. Like, we really need this, but we already have that.
Yeah.
Okay, thank you.
Hey, Mike, Chris Town here. What are you interacting with the Intermine people, because they have a sort of common data model that's shared across quite a number of organisms?
Yeah, so we all yeah, we all have data in it. So that has.
Somebody.
Chris, can you mute?
I'm sorry, I'll mute myself again now.
Thanks.
Yeah, so the Intermine has certainly helped. The majority of the groups have an Intermine. I'm not sure that everybody is at the same level. In fact, in yeast, there's more data in Intermine than we have in our relational database. And actually, there's some things we haven't figured out how to put on the web pages. But that's certainly very helpful. There is an API into that. It's not the solution for everything, but it's certainly been very useful. We're really looking though for a little bit more integration, and this may be more a technical discussion on that. But yeah, they're used a lot.
Thanks. Mike, I had a question kind of along the same lines that Lisa did earlier about your stakeholders, so, or users, depending on your point of view. So one of the key successes of a genome database or genetics database would be the relationship that you have with your users and stakeholders that kind of drives the content that you choose to curate and some of the innovations that databases can provide to provide better access and therefore knowledge for the scientific community. I mean, what we're trying to do is help scientists do their work better, right? So one of the concerns that I think you touched on earlier is if you go to kind of a merging of resources, as it seems like you're being driven to by NHGRI, do you lose that connection to your stakeholders, and therefore does some of the meaning and the utility of the database get lost, and therefore do you actually lose value based on the cost-cutting measures that you've been forced to enforce? So is this a correct assumption that I'm making, first of all? And second, is this something that the funding organizations are aware of, that kind of key relationship that drives the innovation that databases create for the users?
Yeah, that's actually a good point. I think that the tide has sort of turned about the importance of curation. There was sort of a while where everybody thought you could just automate it, or you could have, undergraduates doing it or something, and that would be more than enough compared to really senior, experienced curators. That seems to have turned, and part of that I feel is because of big data. Is that, Lincoln Stein once said, I think it was that big data without small data is junk data. so that you actually need really highly curated data sets to be able to do the right analysis for your big data. And so, and I know I've certainly heard this from a couple of the directors at NIH when they've given talks about things, that curation is very important. So that being said, so they appreciate that we do need to curate a lot of data types, and that many of those data types are more significant to one model organism community than another. I mean, maybe a trivial example is for yeast metabolism, we should really do because it's really well hammered out. And then the understanding of metabolism in yeast is actually applied to many different organisms. So Synerptitis doesn't do much with metabolism, but it's really important that we do. So they wouldn't want us to stop that. There's many sort of better examples of that really had to do with the extent of curation. Would we be more skimming, or do we want to go really in depth? And so that certainly is a concern when we're creating common tools, that we want to allow these various levels of curation to continue. There was just a talk, actually the last hour, among the Alliance all called, that we were talking about various ways of text processing, and then having that lead into some of the curation tools. And so there's some efforts there to sort of try to get that to work. It's already working, I think, for 2 organisms, and it's called Texpresso. I know Tara uses that, I'm pretty sure, or has used that.
I have another question. In your communities, how do you regulate gene nomenclature or even genome nomenclature? And is that a problem? Is that a problem in your communities?
So I don't think it's a problem for anybody. So I think SGD's experience is like the others. There was a nomenclature community when we were founded, and they handed the reins to us, and we're now the authority. So it's up to us to define, the nomenclature. And, everybody's been happy with that. If somebody wants to change a gene name, they have to petition through us, and then we have a process that allows that to happen. If somebody wants to register a new name, we use the word register. A gene name isn't fixed until there's a paper published on that gene using that gene name.
Okay.
But before the paper is published, we allow people to register it, and they register it with us, which makes everybody know that, oh, they're working on it, but they don't have to have any function terms, just that the name is registered.
Okay.
Figure that out. But so as far as genome annotations, as much as we can, we try to follow, really what the bigger genomic community is doing and things. Sometimes these are hard. Our biggest thing, which you guys probably have problems with all the time, maybe you can help me figure out how to do this, is that genes that are in Saccharomyces cerevisiae but not in the reference genome, how do we name them?
Right.
The yeast nomenclature defines effectively that every gene, there's not the gene name, like Act 1, but the systematic name has the chromosome number within the name. We can't do that if it's not in the reference frame.
Yeah. Well, in maize, I work in maize. We're having lots of problems along these lines. So, would you or some people from your group be interested in working with us on some genome nomenclature standardization?
Sure. So yeah, all the mods are the authorities for their organisms.
Okay, for genes, and then for genomes, just like you said, we have now in maize, 12 or 13 reference genomes, and they have don't even contain the same genes. They have different annotation sets, and it's just getting very messy. So we're developing some genome nomenclature, and we thought, this is a problem for everybody. We could do it together and do it a little bit better than we've done for gene nomenclature.
Mm-hmm. Yeah, so that would be something at the mods, connecting with the mods, the people at the mods that do the nomenclature. It's not really an alliance thing, because it really is an organism-based. Nobody's going to try to make the gene nomenclature the same between yeast and fly. So.
Okay, thank you.
So, Mike, Dory alluded to this earlier, but we are in the process of writing a grant proposal towards continuing AgBioData development, and the idea would be to work as a group in 6 different directions, which have some overlap with the challenges that you list on your slides here. So there's definitely overlap in the areas that we're focusing on. Now, you guys are well-established databases. You have lots of funding. You're only 6 of you. And we're a group of like 30 or more databases here of various levels of maturity and depth of curation and data types. We're all kind of related with the ag field, but we're across the taxonomic spectrum as well. So we're seeing here that you guys are having some challenges already in getting your kind of silos aligned, and you're finding ways forward. So considering kind of the demographics that we have within AgBioData, the spectrum of databases, I guess, do you have any advice for us as we're continuing to figure out with writing this grant proposal and with developing AgBioData, like where are the things where we probably will have a really hard time and that we should focus on in trying to do kind of a shared data, shared standards kind of consortium?
Yeah, it's a hard problem. I would say, with the Alliance, we are being successful. It's only like 2 years so far, and things are going better. I guess, so I think I would feel that it's really the data models, maybe one of the more important things to standardize, because once you do that, then everything else can follow. And really, that's the first thing you need to do before you can make a common tools or and curation tools would follow from that. Websites perhaps in the same way. But
can you explain exactly what you mean by data model?
So that you're all representing phenotype. I mean, I think in your white paper you talked about there's a couple different orthologies that are used by some databases, but some don't use an orthology, not a phenotype orthology.
Right. Mm-hmm.
Things like that. So that would be one that, as much as you can standardize that, I think that would be sort of a great point for why it's good to fund the group as opposed to fund the individual projects, because the group as a whole will standardize these things, and that helps all those communities. I mean, that could be one approach for the justification.
All right.
Okay, thanks.
But I think the one way that, at least early on, that we looked at this is that I I made a survey, and I just tried to get some sort of metric on the number of labs that worked on yeast and another one of the mods. So does your lab have funding for yeast and worm, or yeast and fly? And try to get that, because if you do have funding for both, you're using both of those databases. And so that as you'd expect, that number was pretty high, because labs these days are becoming more diverse. And so I'm sure it's probably true for your groups as well. If they're using, Gramine, they're probably also using, I don't really know for sure, but they may be using the soybean or Solanaceae database or something, and so that's a reason for standardization right there, is that would actually help the community. And so that funding needs to go to the group, not to the individuals, because you need to be working together. Maybe that's an approach.
Yeah, good point. I think so far we have not talked at all about like standardizing the user experience either. And that's what you're implying there as well. So that might be something that we'll have to think about.
So I have a question. So on your slide, you listed data types as orthology and human phenotype and stuff like that. And do you have an interface where you can put one of those terms and then see the results from all 6 mods?
Yeah. Okay. So go to the Alliance website, and there's a search box at the top. It doesn't work great for all possible, names for things and data types, but that's certainly one of the areas of development, trying to make that better.
Oh, okay. So it's in the Alliance database, not in each mod, or right. Are you planning to do that as well? Or?
Yeah. So it's really more and more one of the plans is, and I mean, like I say, it's a goal, is to try to create it that general users, people that are not looking specifically for something in yeast, would go to the Alliance page, and that would allow them to see, a particular search for a particular Go term and see the genes that have that function across all their organisms, for example, instead of trying to build that into each mod. That said, we're also trying to build these things as modules. So one of the mods could put that into their database if they wanted.
Okay.
Mike, Ava here. To go back to the shared data model idea, have you built one for the Alliance? And if you have, could you share it with us?
So yeah, so for Go or well, orthology was probably, there were a few. I think we just standardized on one. Phenotypes and variation took a little bit of tweaking to get them to be the same. We have something called gene descriptions, which is really the curated, free text form of how we describe a gene. And this is what we put like on JBrowse or something. That actually took a little while to get nailed down, because that was one thing that was hugely different. So yeah, so there's many of these, and we'd be happy to share them. Yeah. I think some of this is in our GitHub, but it's one of those things. I haven't looked for it, so I'm not exactly sure where others would find it. I just asked somebody for it. Is how I get it.
Yeah. Thanks.
I threw a few of the links into the chat, so they could always follow up that way. Yeah.
Great.
Nathan?
Thanks, Nathan. Any other questions for Mike or about Alliance versus AgBioData from the group?
I'm curious about what the governance structure is, or the decision-making process. When you're faced with a hard choice about which language to use, how do those things get hashed out?
That certainly is. I would say that's been. How would I say it fairly? It's been something that's been a concern for a while and caused a lot of angst with some folks, because, like I say, if somebody really likes Java and they're going to do most of the work, is it fair to sort of have them use Python just because others might know that, but they're not going to do any work? So the governance has sort of been really what the right word is. Not laissez-faire per se, and it's certainly not top down. It's more just sort of a bunch of people running around. It's more mob mentality maybe, but we've tried to empower those that are doing the work to work within committees, and have each committee sort of appoint their own leader, and just sort of see how that bubbles to the top. I think that generally has worked pretty well. I think, interestingly, not to really pick on anybody, but the curators have a much longer history of working together on many things. And so the groups that are really heavy curation, those working on the data, have had less problems like this. And the technology folks are the ones that have done things completely differently. And so that took them a little while to get, I would say, not really fair way to say it, but to get really productive. And but one other thing is that our funding has really been limited, I think, for the technology folks, because that's such a critical person within our own databases that we don't really want to share them as much. But as far as the Alliance, we really needed to share them.
I just wanted to back up what Mike said. The way it's kind of done has been basically who's been able to do the work ends up doing stuff, and it's kind of like you say, self-organized into actually a pretty fairly productive groups. I think on the back end, they've kind of on the technical side, we have groups with a lot of Java experience and a lot of Python experience, and they've kind of different aspects are Java and different aspects are done with Python, depending on who's working on it. On the front end, it's basically been React. So that whole AGR site itself is is done with React as a single page application, which has been, I think they've all been good decisions. Though I might be biased there.
I mean, some of that shows up, and I think some of the, for trivial things like the tables, how the tables look on the page. For some data types, they look one way, and for others, they look some other way. Things are being done, you know, relatively quickly, and so I think the standardization across the whole site hasn't, you know, we just haven't taken time to do that yet, and it's partly because some people are doing one thing, some another, and they use their own expertise, obviously, as much as they can. These technical groups are very small. I mean, in some cases, like 2 people. So.
Any more questions?
I want to follow up again. So it sounds like you've basically moved towards a centralized model where all of the mods are contributing to a single database, and there's a user interface that communicates directly with a single backend. Was there any consideration of a more sort of federated approach where you know, you would continue to remain distributed and autonomous, but there would be interfaces that would, say, call out to services on each of the sites and aggregate them together?
So for a lot of the data, it's really present in all the mods. And so, you know, we need to be consistent data types. So it's not the right use of the word, but each data type may have a separate API. So it's sort of federating across data types. The gene ontology is one case where we're using the gene ontology's APIs, and we're not, well, I guess we're putting some of it inside the AGR database, but not all of it. We've talked a lot about that. Some of it is really just for expedience. Right now, like I say, we're not putting everything from all the mods together yet. It's really those things that it's only the intersection, really. So as we get out to those data sets that are not quite, the really more data types that aren't so similar because of the experimental methods used in the various organisms, those, one thing that we've left ourselves open to is that they'll be provided by the mod groups as an API, and then the Alliance database doesn't need to hold it. So it's sort of a work in progress. I should add, so one of the motivations here is that, so the software is all open, it's available on GitHub and such, and it can be downloaded. It's changing a lot right now, so I don't know that anybody would necessarily suggest that you use it now. But our hope is that with time, we'll be able to include other organisms within the Alliance structure. It's, you know, integrating all together. But I think we also want to make the software available so that others can use that. And you know, you always try to make these things lightweight, but you know, details get in the way of making it light, and it becomes very specific and sometimes a little bit more difficult for others to implement. But at least one of our goals is somewhat like the Gmod model was, that people could download the system, load the data, and they would have a website ready to go. But that's, we're really just trying to nail down, you know, the requirements that NIH has put before us, and so we're not yet talking to other groups. We're not defined other groups that will join. We've been talking to a few and told them, you know, they're sort of high priority, and we want to make sure we stay connected, you know, ongoing, just so that they know how our processes are being defined.
So those data types that you can search from the Alliance website. So are you saying all those data are in the AGR database, not coming from individual mods?
Right, they're loaded into a new database, and it's I mean, maybe can do this more justice, but we use Elasticsearch to deal with the search.
So, but then if you use Elasticsearch, don't the data come from individual database, or
the data?
So, I mean,
right.
Okay, yeah. So basically, that we have loader scripts that go out. So the different mods provide data of a certain type, and those are all loaded in. There's a loader script, I think, written in Python that loads them into the Neo4j database, which is then used. And we have been using Elasticsearch as well to for search and other data retrieval type things. But I think we're moving away from that. I'm a little somewhere in the middle, but it's generally the back end is a combination of Neo.
Moving away from so you're
potentially Elasticsearch. I'm not sure if that decision we're 100% going to do that, because Elasticsearch has been very performant. But we'll see. We'll see what happens.
Okay, so then if you are loading the data into AGR database, you have to be because individual mods, the database is updated pretty regularly, right? So you have to repopulate AGR database pretty frequently, if you want to stay updated.
Yes.
Okay, thank you.
I think it's monthly. Is that correct?
I am not the person to ask, but I think they've talked about doing it between weekly and monthly. Yeah.
Okay. Do you see a lot of users coming into AGR website, more and more, or?
I haven't looked at the analytics recently, but I mean, there hasn't been a huge number compared to the mods, but more people are finding it. And as I mentioned, I had a number of people at the East meeting come up to me and say they looked at the Alliance site and they really liked various aspects of it, and they plan to use it more. So it's getting some traction. Each of the mods has a link on our own gene pages, which goes to the Alliance.
Oh, okay.
And so that's one way that users are at least able to explore the Alliance or learn about the Alliance.
Okay.
Yeah, I think what Suk is getting at is that the model that we've been sort of unofficially pursuing is the federated model. We hadn't even thought about creating an AgBioData database. I'm not even sure that's possible. So it's interesting to see that seems to be the route that you guys are pursuing. Yeah, I'll have to think about that.
We really talked a lot about the federation point, but I think it came down to we would all need to be exporting the same format anyway. So maybe combining it was just an easier way to get there faster. So.
Right.
But Monica, you guys are using Triple for a lot of things, though.
So we did a survey on this, didn't we? And there's a big chunk of the AgBio databases do use Triple, but not everybody. And people tend to use a mix of tools and databases to serve their stakeholders, depending on, for example, Triple plus Intermine, because they both have different functionality. So yeah, but there is a chunk of people who are using Triple. Now, you have to use Triple in the same way and load your data the same way to make that interoperability work, right? So is that what you're getting at, Nathan?
Yeah, I'm just saying it might be more possible because of the similarity between stacks of what you guys are doing. But you might be right, the data types might be so dissimilar that it might make that difficult. I think that the issue with them is we all had developed our stack sort of independently, and so it made doing that type of federation a little more difficult. And some of it was cultural as well, but it's worked out, I think, pretty well. And again, we're mostly centralized, but with some data types like Go and some of the genome data being federated.
Right. Yeah. Well, that's definitely something that we're exploring, and that we'll be exploring should this proposal get funded. So, yeah, Nathan having your feedback there would be really, really good.
Yeah, so I was asking that question because we use Triple databases, and so 3 or 4 of the 3 Triple databases, we use Elasticsearch to search against all the databases involved, and that doesn't really require having a central website to search. So we, from each databases, we just go to connect to other database, and then we can search. So Yeah, but then you do require having the triple for each databases. So.
Yeah, it was just the approach we used. I mean, yeah, Elasticsearch is very configurable. We use Elasticsearch on most all of my projects, other than Alliance 2. So we have some experience with playing with that.
Okay.
All right, looks like we're at the top of the hour. Are there any other short last-minute questions for Mike? Or no? Okay. Well, let's all thank Mike for giving us an hour of his time and sharing all this really, really useful information for us as we're forming this group and figuring out where we need to head out. So thank you so much for taking the time and your advice. And hopefully we'll keep in touch about this.
Great. Well, thank you very much. And thank you for your questions.
Thanks. Bye, everyone.
Thanks.
Bye.
Bye.
Thank you.
Thanks.
Thank you. Bye.
Bye.